an institute studying the molecular mechanisms leading to rapid division of cancer cells. For postdoctoral training, he joined the lab of Professor Robert Malenka at Stanford University, where he studied fundamental molecular mechanisms underlying synaptic plasticity and developed new molecular approaches to study genetic and neuronal networks important for the formation of addiction. So now he joins the ELSC team, and the Citri lab will focus on studying molecular mechanisms of experience-dependent plasticity using addiction as a model. So, please.
Thank you. Okay, so am I heard in the back?
No.
No? Oh, it's open. No. Now? Okay, good. It's a little too loud, no? Okay, so I did recently join. I'm jointly affiliated at ELSC and the Life Science Institute. And we arrived in August after 6 incredible years in California, which I recommend for anyone that wants to leave this crazy place for a few years and get a great life experience. I really recommend California. And we're in the process of starting to establish our lab. What we'll do in the lab is go all the way from the basic molecular interactions through synapses, neurons, neuronal networks, and up to behavior in the whole organism, and we'll try to run along this path back and forth, studying basic mechanisms of experience-dependent plasticity. And our model system is addiction, or addictive behaviors. And the reason that we're working on addiction is that it's just an extremely potent form of memory, and I hope that I'll get that point across to you later on, what a strong form this is of memory. We're going to be using state-of-the-art tools, and I'd love to invite you to see our beautiful lab and state-of-the-art tools. This is what it looks like right now. As you can see, we're really equipped with the best of the best. We're waiting for the demolition team to show up, and we've given them very elaborate plans of what we expect the lab to look like. So hopefully, very soon I'll invite you to see that. So what I'm going to do today is take you on a route, on a path, going really from basic molecular to circuit in the context of behavior. It's going to start a little bit slow, especially for those of you who don't really care about molecules. Bear with me, we'll get there to the stuff that maybe interests you more. And what I want to get across is the methodologies and the approaches, so that you keep in mind where we're going and maybe how you can interact with this lab in the future, how we can develop collaborative work. I'll start from the basic of the basic. Who doesn't know what LTP and LTD are? Good. Okay. So when we talk about synapses, we talk about memory, then obviously we're talking about the models of LTP and LTD, of depressing synapses, potentiating synapses and depressing them. Potentiation is primarily through incorporation of receptors into the postsynaptic membrane. Depression is through removal. And this is what they look like in electrophysiological recordings. You record a baseline, and then you see the potentiation or induced depression. And what I'll describe now is this basic study that looking at the molecular mechanisms underlying synaptic depression that I performed as a postdoc in Rob Malenka's lab. I will not take you through the details at all. It's just, again, so that you have a feel for the approach, and then I'll go into stuff that is the basis for the lab that we're now developing. So LTD, as I said, is by removal of receptors from the synapse. It's dependent on calcium influx through NMDA receptors, but the mechanism through which calcium couples to intracellular retention of receptors hasn't been, wasn't figured out. It's clear that there's an endocytic event that's dependent on calcium, but the identity of the calcium sensor wasn't known, and that's what I set out to study in this project. The system that I used for that is the classic system for studying mechanisms of plasticity. It's the mammalian hippocampus, or the mouse hippocampus in this case. And it's a beautiful laminar structure, very nicely organized, very accessible for viral manipulation, and very homogeneous in its structure, so it's a very good preparation to study basic mechanisms of plasticity. And the other advantage of this prep is that you can move between different levels of investigation, from an acute slice following in vivo manipulation to slice cultures you can hold in culture for weeks, and dissociated neuronal cultures, and these different systems complement each other very well. So setting out, I quickly zoomed in on this protein, PIC1, and again, I won't go into detail. If anybody is interested, we can talk about this later, why I chose this molecule, but it had domains that are interesting in the context of long-term depression, potentially. And the approach that I took was one that was developed in the Malenka lab, which is molecular replacement using multi-promoter lentiviruses. The idea here is that you insert an shRNA that knocks down your protein of interest, and then you replace that protein with a recombinant form that's tagged with GFP. And what this allows you is, and here you can see this is the endogenous levels. We knock down the protein, and then we replace it to very similar levels to the endogenous with a tagged form. What this allows you to do is detailed structure function analysis of the protein and address its function in the phenomena that you want to address. And the approach that, the context we do this in is, again, in hippocampal slices and performing paired recordings, dual whole cell patch clamp recordings, where we record from an infected cell and the adjacent uninfected neuron. And this is actually the best controlled experiment you can perform in this context, because these cells receive very similar inputs on average, and therefore you're only studying the modification due to the viral expression. So the first question, obviously, is whether this protein is essential for LTD. So here you see in controls the induction of LTD, and following knockdown of PIC1, you can see that you cannot induce LTD anymore. Then the second question is whether we can rescue this LTD by expression of a recombinant form. And here you can see that you can rescue LTD fully if you replace the endogenous with the recombinant form. What this shows is two things. First, that PIC1 is essential for LTD. And secondly, that the system is a valid system in which to address the function of PIC1, because we can do this molecular replacement that works effectively. And now we can go in and study this molecule in detail. So as I said before, the advantage of the hippocampus is you can move between these different levels of analysis which complement each other, and we can study receptor trafficking in dissociated neuronal cultures, where if you treat them with NMDA, you see the endocytosis of AMPA receptors. If we knock down PIC1, then we don't see this intracellular accumulation of AMPA receptors. But if we replace the endogenous protein with a recombinant form, then we can rescue back the normal phenomena. So we have a nice complementary system in order to look at the trafficking. And now I'll spare you all the details and just run to the conclusion, which is based on very detailed structure function analysis of PIC1, together with biophysical and biochemical measurements. And the model we came up with, I think, is very well substantiated by the data, and it looks like this.
But I'm sorry, but you're talking about LTD and you talk about increase in AMPA receptors?
No, the increase in the intracellular accumulation of AMPA receptors. Not on the membrane, but the, yeah.
Oh. But you don't have an explicit measurement of the surface?
We do. No, we're looking both at the surface receptors and at the intracellular receptors. We're looking at the ratio. You always need to, in those experiments in particular, you have to do a normalization of the internalized versus the surface as a way of controlling.
Totally is more or less
No, that's why you need to normalize for each cell that you look at. You need to normalize for the total amount of receptors because it does vary. So that's why you're comparing the endocytic fraction versus the rest. But that's only for this particular assay. So the model is that following calcium influx through NMDA receptors, we have a internalization of AMPA receptors which is independent of calcium, an initial event that's calcium independent. And then we have a calcium-dependent modification of the structure of PIC1, which now meets the receptors in a endocytic vesicle and inhibits their return to the membrane following the endocytic event. So effectively what you get is a reduction in the number of receptors at the synapse and their accumulation in the cell. And if anybody is really interested in the details, we could discuss them later. Now I'd like to take you to the model system that is interesting for us and forms the basis of work in the lab, and that is addiction. And addiction is, it's not commonly appreciated, but addiction is the most dangerous in terms of human fatalities, the most prevalent in terms of number of people affected, and the most costly of all neuropsychiatric disorders. So this is a major disorder affecting society very severely. And it's, I like to view it, and a lot of people, I'm not alone here, like to view it as a disruption of memory, just an extreme form of memory that causes you to lose control over your behavior. You create a very strong association that takes over your behavior and leads you to behave in a way that you can't modify anymore. And therefore, the idea is that this would leave a very strong trace in your brain. It would modify the brain in a way that we can now look at it and dissect the mechanisms underlying it, and that's why I feel that it's a very good model for looking at experience-dependent plasticity. And just to clarify, by experience-dependent plasticity, I mean the fact that you have an experience like listening to this talk. This modifies your brain in some way and hopefully changes the way you think about things in the future to some extent. So we have neuroplasticity and the neuroplasticity affecting behavior.
You made several assumptions that I'm not sure but you said that it affects memory, that there's obviously addiction or compulsive behavior which is working against memory. I mean, you remember everything but you still do it.
No. What I mean here is that, I guess I'll clarify it later on. But
because other things other than memory affect the brain. The other traits which are not memories per se. Okay.
Oh, maybe the nuanced meaning of what I say will be clear later on. So why is this a good system to look at experience-dependent plasticity? The advantages of studying the reward circuitry in the context of drugs of abuse are that addictive drugs elicit robust and reliable behavioral plasticity that is very stereotypic, and also you can see modification of synaptic function that is also apparently reliable. I mean, labs can replicate each other's results, which is always a nice advantage in the system. The animal models for addictive behaviors reliably replicate the human condition, and this is by far more true for addictive behaviors than for any other neuropsychiatric disorder. And the coarse circuitry has been defined, but it's been defined in coarse terms. So we have a nice structure to work on that's already been established, but there's room to find new, to make new findings here. And from our perspective, it's acutely inducible, meaning that as an investigator, we have very good control over the timing. And this is crucial for the type of experiments that we want to perform. So just to give you a flavor for what addiction is about and how we view it, and maybe also to begin to answer Tali's question, I'll share a brief story that a Swiss colleague likes to tell, and this is about a man who's a successful businessman, has a nice family life, everything is fine and dandy, lives in a nice rural environment, But in his past, he was addicted to cocaine. And this person was living his life well until he moved to a new environment. And then what happened is that in this environment during the harvest season, he would be driving back from work and everything would be fine until at some point in his route he'd be driven to go get cocaine and revert back to his old behavior, his old addicted behavior. And he couldn't figure out what this was. This was destroying his life. So together with his physician, he went over the route. And what they realized is that everything is fine during most of the route, but then when he hits this field with these bales of hay covered in white plastic, the white plastic triggered in him this memory of these other white plastic bags, and this immediately reverted him back to his behavior, his addiction. And what I think this gets across is the potency of this memory trace that I'm talking about, that this man is bearing. It's there, it's hidden. He can normally live his life, but once the trigger shows up, that's it. He's done. And this is something that I think suggests that this is a good place to study mechanisms of plasticity. There's something hardwired into the system, and maybe it's amenable to description in a good way. And hopefully the secrets to that are hidden within this circuit map, or maybe modified versions of this circuit map. So the basic, the central element of the reward circuitry is the nucleus accumbens, which sits in the ventral part of the striatum. It receives multiple inputs from different brain regions and creates an impression of how rewarding an event is. That's maybe in rough terms the way it could be described. The inputs that it receives are from the prefrontal cortex, which gives an evaluation of, which is executive control of function, again in rough terms, and contextual information it receives from the hippocampus, and then information about emotional or stressful levels of the organism come from the amygdala. All these inputs are glutamatergic inputs, and glutamate inputs into the accumbens have been shown to be relevant to addictive behaviors. And each of these brain regions, through ablation studies, has also been shown to be relevant to the development of addictive behaviors. An additional crucial input is that of dopamine from the ventral tegmental area, where dopamine provides the information regarding the valence of the activity, the rewarding level of the experience. And being the central integrator of signaling and reward, this is where our basic interest lies, in studying the mechanisms in the nucleus accumbens. Zooming into the nucleus accumbens, or this is true overall for the striatum, 95% of the neurons in the striatum, and specifically in the nucleus accumbens, are medium spiny neurons that you can divide into two types, D1 and D2. The D1 receptor expressing medium spiny neurons belong to the direct pathway, meaning that they directly synapse on the VTA, and the D2 receptor expressing medium spiny neurons are indirect pathway neurons that synapse onto the ventral pallidum, and then the ventral pallidum synapses onto the VTA. Functionally, it's thought that D1 expressing cells are encode an active function, whereas the D2 expressing cells are antagonistic in their function and reduce the development of a behavior. And this has also been implied in addiction for the nucleus accumbens. On the other hand, though, when you look at this, and this will become relevant later, it's also known that cells in the striatum fire independently of each other. They don't fire in a coordinate fashion. And it's been suggested that they function as ensembles, that ensembles are recruited to subserve a specific function, meaning that when we perform analysis of this whole brain region and electrophysiological recordings of specific cells, we're likely missing the bulk of what's going on, because we're not recording from the cells that are specifically involved in the behavior, but we might be missing the real action. And this is relevant to what we'll come to later on. So the basic approach that we'll be taking in the lab, and this appears like hand-waving at this point. It is hand-waving, but it'll give you structure to what I'll show you later on. We begin by studying the molecular network in the nucleus accumbens that's responsive to cocaine experience. We choose genes, target genes, for functional analysis. These genes, their analysis feeds back and leads to higher resolution in our understanding of the molecular network. From the understanding of the molecular network, we can also create novel tools to decipher the neuronal circuits. And this is a major point that I will get to towards the end of the talk. The new understanding of neuronal circuits also can feed back and enable us to get higher resolution understanding of the molecular network. And in this way, we have an iterative process where we can increase our knowledge with each round of the functional circuitry in the nucleus accumbens, both at the molecular and synaptic level, and hopefully get a better understanding of what underlies the transition to addictive behaviors. Now, the history of all this goes back about 7 or 8 years to cancer models. And this, during my PhD, I was studying the response of epithelial cells to stimulation. These are human breast epithelia. And they respond completely differently to EGF or serum. If you dump EGF on these cells, they undergo migration. If you put serum, then they undergo proliferation, meaning two completely different cell phenotypes following different stimuli. When we look at the transcriptional program induced in these cells, it looks similar in terms of its temporal organization, meaning we see waves of transcription. We see the small, very early wave, and then a second wave following that, and we can cluster this into a third wave, and then slow accumulation of additional proteins, and this holds true for both programs similarly. But when we compare the identity, and I'm sorry, those of you that aren't familiar with gene expression analysis, we're looking here. Blue is low levels of expression, red is high levels of expression. Each column is a time point in the experiment, and each row is a single gene. And when we compare the identity of the genes between these two programs that send the cells to different fates, what we see is that they only share about 40% in the identity of the genes, meaning that a similar structure, a similar dynamic is used repetitively with different identities of the underlying components. And the different identities have also a stereotyped organization to them. What we see early on is genes that promote negative feedback of signaling. We see transcriptional regulators, master regulators of transcription, modifiers of transcription, RNA stability proteins. All these together we can define as performing a computational event. They're taking the stimulus from the environment and they're defining what is going to happen. They're the ones that are responsible for computing how the cell is going to respond. And how the cell is going to respond is encoded by the genes that are induced later. Earlier, at these time points, we see genes that are involved in propagation of the signal, genes that are perform cell-cell communication and cell-cell interactions. And later on, we see genes that are very specific to the event that's been induced. So genes that are involved in changing the cytoskeleton, genes that are involved in cell matrix interaction, you'd see in cells that undergo migration, and cells that are proliferating, you see the appropriate genes for cell cycle. Now and then we can go into even higher resolution where we can see how transcriptional complexes are organized a long time, and there's a logic to that, and we can identify specific genes that are responsible for specific phenotypes, such as in this case, this protein C10 we implicated as a major protein responsible for cell migration in response to EGF stimulation. Now, why did I bore you with this whole long discussion of transcription? It's because it's relevant to how neurons respond to stimulation. This appears to be a basic structure of how cells respond to stimuli, and neurons are no exception to that. Meaning when we look at the response of neurons to stimulation, and it doesn't matter what stimulus they see, the response is structured in the same way. We have an early computation event where we have all these different components that I told you about before, and the delayed response where we see proteins that could be involved in performing the function itself. Okay, now the experimental system that we're studying is the simplest model for cocaine response, in vivo cocaine experience. This is the development of behavioral sensitization to cocaine, where we take a mouse, begin injections with saline in a novel environment while measuring its locomotor activity. And then you can see it habituates to the injections and the new environment. And then it experiences cocaine for the first time by an acute injection of cocaine or daily injections of cocaine, which is termed chronic exposure to cocaine. Now, after that, we stop giving the animal cocaine. And this we can do for days, we can do it for weeks, we can do it for months, it doesn't matter. When you come back, and this is termed abstinence, a period of abstinence, when you come back and give the animal what's termed a challenge dose of cocaine, you can see that the response is elevated to the level of the last response to the chronic exposure to cocaine, and many times above that. And the longer you wait, actually, the higher chance you'll see an elevated response. And what you see here is, again, this strong memory trace that you have in this animal where you see this very potent memory of the response to cocaine. Again, suggesting that this is a good model in which we can study the underlying mechanisms.
What is the behavioral response that you measure?
It's just locomotor activity.
It runs around?
It runs around, yeah. And we also have other models, but this is the basic model that's the basis of most of the studies. Within each one of these time points, we study the transcriptional response, the dynamics of the transcriptional response to an acute cocaine exposure or following chronic cocaine or to a cocaine challenge. And what we see is that overall, although it's not as elegant as in cultures, overall the response is again stereotypic, and we see the same signatures that I described to you before, where there's a logic that associates between gene expression along time and gene function. Okay, and now I'll tell you the next few slides. I'll just run quickly through some initial interesting observations which will form the basis, each one of them, for a separate project in the lab. And later I'll get to projects that are a little bit more developed and perhaps more interesting to more of you in the audience. When we compare the response to acute cocaine to the response to cocaine challenge, we see that both of them have this temporal structure. When we look at the identity of the genes induced by acute cocaine, now we're looking at them within the data set of the cocaine challenge, we see that they're not induced, that it's a complete mess, meaning that it's not the same gene program induced by acute cocaine and cocaine challenge. When we look at the genes induced by cocaine challenge and look in the data set for acute cocaine, the reciprocal comparison, we again see that there's no structure there, meaning that the identity of the genes is completely different. There's a complete rewiring of the transcriptional response to cocaine due to this cocaine experience. So this is experience-dependent plasticity at the molecular level. Something there is really changing the response and perhaps underlies the difference between these animals, an experienced animal and a naive animal. And we have reason to think that a lot of this is epigenetic, and we're going to address this in detail.
This is the response to cocaine exposure after acute cocaine or after cocaine challenge?
No, yes, you give the animal, you take a series of animals and you give them an IP injection of cocaine. The first one, you take its nucleus accumbens after an hour. The second one volunteers its accumbens after 2 hours, and so forth. And then you look at the development of the transcriptional response a long time. That's the idea. Either following one single dose of cocaine, that's the acute response, or after the development of the chronic response to cocaine.
So after cocaine challenge, you give a cocaine dose and you measure the response?
Yeah, that is the cocaine challenge, is this dose of cocaine given after this period of abstinence. And then we're looking at these time points after that cocaine injection.
Does the pattern matter? I mean, you call the first stages the computational stages, and they seem to be bigger in the acute challenge, the acute cocaine, and then it's like they skip this computation and run to it.
It is, it's evident in the data that really, in terms of those genes whose identity will, like transcription factors, genes that are involved in all these genes that modify signaling and all that, that component is extremely small. That's what we're going to try to figure out why and how that impacts on the behavior of the animal. That's the point of this particular project. Then another interesting thing to look at is to compare naive animals immediately before this acute cocaine dose to abstinent animals, those that are immediately before this challenge dose of cocaine. And you can see that there's a change in a large number of genes. But there's a subgroup, relatively small subgroup of about 30 genes in here that looked extremely interesting. And the reason that they looked interesting was that they were upregulated, they were completely absent in naive animals, upregulated enormously in abstinent animals, and then following this challenge dose of cocaine, they immediately disappeared again. And this is a peculiar phenomena in biology. I mean, this could happen because of RNA degradation, or it's not a standard thing, and especially not given the levels. But looking at the identity of the genes, there was something common to them. A lot of them are immune genes that are completely foreign to the brain. And trying to piece this together, came up with a model that this might be some type of immune cell, that there might be some type of neuroinflammatory process going on here following the chronic exposure to cocaine. There's a little bit of evidence in the human biology of cocaine addiction suggesting neuroinflammatory processes, but really nothing there. And starting to look at this and looking at markers from here, then we do see cells that are found in blood vessels that label with markers from this group of genes. And it will be very interesting to see whether there is a neuroinflammatory component to the development of the response to cocaine. No, this is just the nucleus accumbens.
Yeah, but can you, of the whole brain tissue, or you can limit yourself to profiling neurons?
Not currently. We plan to do that in the future. This is just taking a hunk of tissue and profiling what's happening there. So my current thought is that there might be a recruitment of some immune cell that, due to neuroinflammation to this region, it might have a role in maintaining the elevated response here. It could be a completely new avenue to study addictive processes. Now I'll get more to the meat of what we're really going to look at in the near future in the lab, and this is studying one molecule that's induced following an acute cocaine exposure. This molecule is called EGR2, And you can see here its induction profile. This is actually an extremely dramatic induction, especially for an in vivo situation where we're profiling a hunk of tissue, but actually the gene is expressed only in select cells. You can see it's a classic immediate early gene. It's induced very quickly and decays very rapidly to below baseline levels. Also, I hope this helps you get an impression for the quality of the data. This is in vivo transcription, and this profile is extremely clean. Each time point here is a different animal. So this, I hope, gets across that we really can study transcription in this system. EGR2 is a regulator. It's a transcription factor. It regulates transcription. It's been implicated as a negative regulator of transcription, which will be relevant soon. What we do is take adenoviruses, put in a GFP cassette and a shRNA cassette, inject them stereotactically, and we can see effective knockdown of EGR2 by at least 80%. And here you can see that we label the nucleus accumbens very effectively, and it actually is restricted pretty well to the nucleus accumbens. Looking at the behavioral response to cocaine experience, again looking at locomotor activity in behavioral sensitization, we can see the classic development of the behavioral sensitization, and following the knockdown of EGR2, what we see is actually an elevated response. And this elevated response suggests two things. First of all, it suggests that the animal is normally limiting its response to cocaine, which actually makes sense. And it also fits in with the idea that EGR2 might be a negative regulator of transcription, and it suggests many, many future experiments to study how EGR2 could be functioning in the system. One of these is to perform, as I described to you before, the dual whole cell recordings from infected neurons and uninfected neurons, and address the role of EGR2 in the gene program that leads to modification of synaptic efficacy. And this we can do either with electrical stimulation or with optogenetic stimulation, where we look at a specific input, let's say from the hippocampus, and correlate it with what's known about the change in synaptic efficacy during the development of the response to cocaine. And this is just the basic scheme of how we plan to address molecules of this type in the system. We start off by developing an shRNA, inject it in vivo, look at the cocaine-elicited behaviors, perform electrophysiological recordings, look at the impact of knockdown on the gene on the transcriptional program, and then refine our understanding of the transcriptional program by defining the downstream target genes, which we can then introduce again into the scheme. And hopefully, downstream of EGR2, we'll be able to isolate a molecule that is responsible for the change in receptor levels at the synapse specifically, but that is for future work. Now, what was interesting looking at the induction profile of EGR2, it suggested that maybe the gene is induced to very high levels in a small subpopulation of cells. As I said before, the accumbens doesn't work in a concerted fashion, but it probably works as ensembles of neurons. And maybe we could use EGR2 to mark these neurons that are responding to cocaine experience. So by immunofluorescent labeling for EGR2, you can see that it co-localizes preferentially with the subpopulation of D1 neurons. And D1 neurons, as I said before, are those that are more implicated in driving the behavior forward, driving the development of the response to cocaine. To address this, we used a mouse system, a transgenic mouse developed by Patrick Charnay in Paris, where he knocked in Cre into the EGR2 allele. So wherever EGR2 is expressed, Cre will also be expressed in the heterozygote mouse. Into these mice, we inject an AAV virus, which has a double-floxed inverted GFP cassette, meaning that this virus doesn't express GFP normally. But only, and then we inject these into the accumbens of mice. Only following cocaine will we have induction of EGR2 or Cre. Cre now recombines this cassette and causes GFP to be active. So now we're encoding GFP in the cells that have responded to cocaine, and indeed this appears to work effectively and label these potential neural ensembles within the accumbens. And this is, again, the basis for another project in the lab where we'll study this neural ensemble in detail, and also, as Abdi was alluding to before, we could also look at transcription from within these neurons in a selective way and study really clean out the response to cocaine and look only at the neurons that are potentially involved. And now this is the last story that I want to tell you today, and it's the one I like most up till now, because it's a nice story in science about how you go and look for something and you're focused on the target in front of you, but you always should keep your peripheral vision open and be ready for surprises. And what happened here is that I used to inject the virus into the accumbens, and then I had reason to inject it a little bit more laterally. And what I saw is this streak of neurons that was very reliably labeled. Every time I injected here, I saw the same labeling pattern. And looking at the identity of this, it turns out that this brain region is called the claustrum. I didn't know what the claustrum was. But the claustrum is a very fine streak, paper-thin streak of neurons sitting sandwiched between the striatum and the cortex. And it is very similar in structure in humans, as seen here, or in mice. And there's very little literature about the claustrum, probably because it's extremely difficult to study some paper-thin structure that sits sandwiched between two major brain regions. But the little literature that was there made these two people very excited, Francis Crick and Christoph Koch. And in a paper that they were editing, Francis Crick was editing on his deathbed. So these are actually the last words he left to the world. In terms of science, they thought that this brain region could be involved in consciousness. They were intrigued by finding where the soul sits in an individual. A pretty lofty idea. And they based that on the literature suggesting that the claustrum is involved in modality integration, that it takes inputs from all the different modalities and integrates them in one place, and there perhaps that's where we sit in the brain. I have, based on the literature, I have a slightly modified version of that, which is a little bit more modest, but I think still very interesting. I think maybe a little bit more along the lines of attention, but we'll have to look at that and see if that's true. The claustrum receives input from almost all regions of the cortex, and it projects back to almost all the regions of the cortex. They said that it has a homogenic structure with relatively few neuronal types and a uniform architecture. I hope now we have the tools to look at that in more detail and see whether that's true. And it was impossible to perform conclusive lesion studies or targeted in vivo recordings due to the structure of the striatum, due to its anatomy. And what they suggested in their paper is that molecular tools are necessary in order to test the function of the claustrum, and they ended with memorable last words: what could be more important, so why wait? So driven by Francis Crick and Christoph Koch's, Francis Crick's dying words, why wait? So set out to address how the claustrum might be functioning in the response to cocaine. And here I applied another cool molecular tool, which is a potassium channel. If you express a potassium channel in neurons, you're going to hyperpolarize them, and they won't be able to fire. These are controls. These are neurons expressing the potassium channel. They won't be able to fire to the same extent. They'll be effectively knocked out of the circuit. Putting this in the context of this transgenic mouse, then only following Cre expression, we'll get expression of the potassium channel, and we'll label the neurons with GFP. And what we see is that control mice express the stereotypic response to cocaine, behavioral sensitization to cocaine. But mice which were bilaterally injected with this double-floxed inverted potassium channel initially respond to cocaine, most likely due to the fact that there's a delay between the cocaine experience and the time it takes to produce the channel and send it to the right place. And then the response drops down to basal levels. This implicates the claustrum as a component of the reward circuitry in the context of cocaine experience. And now I feel a pretty heavy onus on my shoulders to see how we can really demonstrate that this is true and associate the claustrum with the reward circuitry. For this, we're beginning by using another nice molecular tool, which is rabies virus. Rabies virus in the wild acts by getting inserted into the animal peripherally by being bitten by a rabid wolf or fox, let's say, and then it's bitten peripherally. The virus goes into the peripheral nerves and runs along the axon and hops back synapses until it reaches the brain. Using an attenuated virus, you can stop it from running back, and you can actually look at one synapse back from the cell that you're interested in, and therefore map the circuitry. And adding on an additional layer that allows you to target infectivity of rabies virus only to the cell population that you're interested in, then you can look at the circuitry onto your population of interest, and that's what we applied here. And the idea is that we label claustral neurons with a red fluorophore TD tomato, and the rabies virus labels the presynaptic inputs in green. And indeed, this appears to work very effectively, where we have three different ways of sectioning the brain: coronal, sagittal, and horizontal, and you can see the labeling of the claustrum. You can also see what a fine streak of cells this is, and you see the presynaptic inputs. Here again, claustrum and the presynaptic inputs. And we're in the midst of analyzing this in order to be able to give names to the inputs and perhaps put them in some context.
Maybe how can you differentiate between the claustrum and the accumbens in all these manipulations?
This is all injected laterally. It's injected into the claustrum. The accumbens would sit here. So that we target the injection laterally, and that's just by... just to give you a flavor for some additional tools that we plan to utilize in this context, I showed you the potassium channel that we used, but we're planning to use optogenetic tools to look at the function of the neurons that interest us both in the claustrum and in the accumbens. And then to gain higher resolution, I showed you what beginning to do with rabies virus, but we also plan to look at expression within the target population using a tagged ribosomal subunit, and then we can profile the active transcripts from within the population of interest, and also perform conditional shRNA, create a conditional shRNA, which allows us to look at the function of genes from within the population without affecting surrounding cells. So I'll just reiterate what I was trying to get across, the idea of how the approach that we're taking in the lab. Begin by dissecting the transcriptional program. Liat, a ELSC-funded postdoc in the lab, will be responsible for carrying this project forward. We'll perform target identification and investigate the function of specific target genes, and Hagit, who is lab manager, hopefully will have the time also to perform some scientific investigation once we get the lab running, and she'll be responsible for promoting this project for EGR2. And as I said, also we can develop tools to identify neural circuits and investigate their function. And Yael Gol, who's starting her PhD in the lab, will be responsible for the claustrum project. The last point I want to get across, I think this is the last point, is that we're in the midst of two major revolutions in biomedical science. One is the genomic revolution. This is the one that you might be less familiar with. And this is a technological revolution where we're daily getting new tools that allow us to look at genomes with higher resolution, reduced cost, and understand how they function better and better. And this heralds in the idea of personalized medicine, where we can understand the basis for disease on a personal level and target the therapy appropriately without side effects and in a more effective way. And it seems like this is a dream, but it's actually really coming true. And this is a very dramatic technological revolution. The other one you're much more familiar with, and this is the neuro revolution, which I see as more of a sociological revolution, where society has decided that we are mature enough to understand how the brain could function, and we deserve to be funded and to develop the tools to address this, and also that we have a huge responsibility to resolve psychiatric disorders which have fallen behind and haven't been, there are no new therapies really for psychiatric disorders coming up in the past few decades. I'd like to thank you for your attention. Say that this work was performed at Stanford. I was funded by these agencies there, and now funded mostly by ELSC and these other agencies. Thanks. One last ad to add in there.
Thank you, Anya. Questions?
So I'm going out of the seminar. I learned something. It will stay in my brain for a while. Do you think these are
Relevant?
Yeah, these are relevant also to this kind of plasticity?
My hope is that mechanisms of plasticity are redundant throughout the brain. That's my hope. It's true for synaptic plasticity that you see different types utilized in different brain regions. But you do, like NMDA receptor-dependent mechanisms of plasticity, I think you see throughout the brain. I don't see brain regions that don't utilize these mechanisms. And I think that much of what's true for one brain region will apply to others also in terms of the molecular mechanisms.
How is the mystery of synaptic specificity, you think, is resolved? If it is not an addiction or preconditioning, but more specific information, current thinking is that the specific synapses are strengthened or weakened. How the machinery of the transcriptional network is
how does it know to, yeah. There are some ideas for molecular tools that would allow you to maintain efficacy at a given synapse or target changes to a specific synapse. There are molecules that have been suggested to have this function.
But your lab is not going to address this question in the next 20 years?
No. We won't. It's a very challenging one. The resolution is so fine that you really need to be able to look at the single synapse level in the behaving animal to really address this.
But that's what ELSC in principle gives you. I mean, in terms of the possibility of proof.
Okay, so that's why I wanted to present myself and hear the ideas of how we can do this. Right now, I don't see it in the scope of what we'll do, but I'm open to suggestions, definitely.
Any more questions?
I have a more specific question. potassium channel that prevents the cocaine response? It's a cell-specific introduction or the global?
No, it was a conditional expression only
so
within the.
What I know, and this guy used to tell me, that the system will make all effort to keep
compensate
balance, the
for
inhibition and then homeostatic. How come you got a response? A reduction in the response to cocaine. And it's not acute, it's a long term, right?
Yeah.
It takes time. I think that the system is built so if something goes down, it will do all the effort to bring it up again, and then.
But not for cocaine response.
No, but
not
specifically it will respond differently. How come?
No, I think that there are two levels of addressing that. One is the level of at the single cell level, whether if you introduce a potassium channel, the cell can compensate for that potassium channel and will fire now in response. No, they don't fire. I mean, you can ablate them from the system effectively. The question is now whether you can transfer the information that was supposed to be encoded by that cell to an adjacent one, and I don't see evidence for that. That's all I feel safe saying.
Potassium injection is not just excitatory, it's also inhibitory now.
No. This is only within the population responsive to cocaine. I can't tell you. What I can tell you is that EGR2 is the very highly co-localized, the EGR2 expression with the D1 population in the accumbens. Actually, no, that's not relevant to this question. Here we're looking at the claustrum. We have not looked at all at the identity of the cells. I don't know what percent of the claustrum we're affecting, what the identity of the neurons are. I have no idea.
But when you say
this we need to start addressing.
You mean to the application of cocaine? Or just don't fire, period?
No, it's just very hard to drive them to fire. I don't know. I don't do in vivo recordings. I don't know effectively what goes on. I know that if you model it, or if you take slices and look at cells that were injected with the potassium channel, then they're hyperpolarized and it's harder to drive them to fire.
This is what you're doing optogenesis, so you can turn them off in real time.
Yeah.